Hello, everybody. Good morning, and thank you for joining us for the ALBA-Elsevier Award Lectures on Brain Sciences. I'm Megan Carey. I'm the past chair of the ALBA Network. And ALBA is committed to promoting equity and diversity in neuroscience. We engage in a range of initiatives aimed at tackling underrepresentation of women, racial and ethnic minorities, and members of other underrepresented groups. We're a European-based organization with a global reach, which reflects our close relationships with our founding partners, FENS, EBRO, and SFN. One aspect of our work focuses on geographical and global diversity, which we're highlighting today with this award. The ALBA-Elsevier Award Lecture on Brain Sciences recognizes research achievements of outstanding quality by an active scientist working at the frontiers of brain research at any career stage post-PhD or MD from countries that are underrepresented among neuroscience publications and conferences. So the goal is really to highlight the awarded individual's scientific excellence, as well as to support regional diversity and increase the visibility of both the scientist and their host institution. And we do this in part by providing a platform for them to share their science at major international conference like FENS. So today I'm very proud to announce the winner of the 2021 award and introduce Professor Seung-Hee Lee, who's an associate professor at the Korea Advanced Institute of Science and Technology in Daejeon, South Korea. Dr. Lee has received this award in recognition of her major discoveries in neuroscience, particularly in unraveling the neural circuit mechanisms underlying multisensory integration and perceptual decision-making in mammalian cortex. Dr. Lee received her PhD in biological sciences and neurobiology from Seoul National University in 2007. Following postdocs there and at UC Berkeley and the Howard Hughes Medical Institute in the laboratory of Yangdan, Dr. Lee established her lab at KAIST. Professor Lee has led her team to significant discoveries, including the selectivity of neuromodulatory projections to sensory cortices, neural circuits for auditory dominance over visual perception, mechanisms through which somatostatin enhances visual processing and perception in visual cortex, as well as gated feed-forward inhibition and its role in the frontal cortex in goal-directed actions. Besides her research achievements, Professor Lee has also contributed towards promoting the neuroscience community in South Korea and ensuring that research from this region reaches a wider global audience. She's received many awards for her work, including the Young Scientist Award by the World Economic Forum, 30 Young Scientists of Korea Award in Life Sciences, and the SciTech Korea Young Neuroscientist Award from the Korean Society for Brain and Neurosciences, among other honors. So today, Dr. Lee will present her work on the neural basis of sensory processing and multisensory integration for decision-making. So please come up and join us. Thank you. And just a note to say we'll be having the formal award ceremony at the end after both lectures.
Thank you for the very nice introduction, and it's my great honor and pleasure for me to be here. And I'd like to thank the ALBA Award Selection Committee for giving me this great opportunity today. So today I'm going to talk about our recent findings on neural circuits for multisensory integration and perceptual decisions. So let me start with this philosopher. I guess you may know about him, Arthur Schopenhauer. He wrote a book, The World as Will and Representation, on 1818. And this book starts with the first chapter with this first sentence, the world is my representation. So this sentence, apparently he noted that the world that we understand is not the physical itself, it's the representation in our brain. Not only the philosophers, but the psychologists also understood that the world is our representation in the brain. One example is the McGurk effect. So McGurk effect is the illusory percept formed in our brain when we receive the conflicts of the auditory and the visual inputs during speech understanding. So McGurk and MacDonald, the psychologists, did this test in humans, giving the sound stimulus ba together with the auditory stimulus ga, and our brain naturally tried to resolve the conflicts between the auditory and visual stimuli and generate the fusion response da in the brain. So many of us experience this. So let me test this with the movie that I made from my students. So please see his lips, what he's saying, to see what he's saying. Do you hear ba or da? Da or ga, I guess. So let's close the eyes and listen it again. It's the same movie. So now you can probably hear the ba, right? Without the conflicting visual cues, we can hear the ba clearly, but if we see the lips saying ga, then we somehow make this illusory perception that is not existing in the sensory world. So how our brain make this illusory percept, right? And actually in individuals, there are variations in this mechanical illusion experience. Some people show very strong experience of the mechanical illusion, but some people still hear ba even though there is a conflicting visual stimuli together with the sound stimuli. And in the adults, more than half of adults experience this mechanical illusion, but many of the children still, more than half of the children show the auditory dominant perception, indicating that learning and experience may influence on this McCurk effect. So how our brain can generate these variations in the McCurk multisensory integration of audio-visual conflicts is still unclear, especially when you think about the complicated circuits in the brain and how this interpretation happens in the brain circuits. So philosophers and psychologists understood that the world that we understand is the representation in the brain, but still in the modern neuroscience, it's still unclear how the brain represents the world. So somehow in the brain, different sensory inputs get together, and within these complicated circuits, the sensory integration happens, and the brain can extract the subjective meaning from the stimuli, and then it can guide the optimal and flexible decisions in the behaving animals. So how this happens is our major question. So people try to find such a reason that integrate different modality inputs as a multisensory integration areas, and found there are cortical regions that can respond to different types of sensory stimuli, and they named it as an association cortex. And they found that there are multisensory neurons that show stronger response when there are coexisting different modality inputs, like auditory and visual cues are present together, and this neuron can show the stronger responses than the unisensory, to the unisensory stimuli. And also people did the brain mapping and tried to identify the specific regions that show the multisensory responses. And as you see marked in the red colors here, these regions are the regions that shows the responses to all auditory, visual, and tactile stimuli. So there are association cortical regions that respond to the multisensory stimuli. And also, it is found in the primate brains as well. There are various regions that show multisensory responses. But in the cortex, within 1 cubic millimeter of the cortex, there are more than 50,000 neurons. And each neuron, as you know, it receives more than 6,000 synapses. So how these complicated circuits can integrate the multisensory inputs? And within the cortex, there are different types of neurons, excitatory and inhibitory neurons. And in the inhibitory neurons, there are different types of neurons defined by the genetic expression patterns and also the microcircuit regimes. But still, we don't understand how these cell types and circuits make sensory integration and guide the animal to make the optimal perceptual decisions in the multisensory environment. So we wanted to test that we wanted to find that in our research. So to find that, we used the mouse model. So the mouse showed the perceptual behaviors like the humans and the primates. And we devised the mouse audiovisual discrimination test to train the mouse to discriminate both visual information stimuli and the auditory stimuli. So we usually use the oriented bar stimuli as the visual cues and the two tones for the auditory cues. And we train the animal to understand a set of visual and auditory cues as the go or right choice stimuli, and the other set of the visual and auditory cues as the no-go or left choice stimuli. So we randomly present these 4 types of stimuli to the animal until they learn the meaning of each stimuli well. And after the training, we can test the animal by giving either the congruent audiovisual stimuli, like giving the auditory and visual stimuli as they show the same meaning in the stimulus, or we can give the incongruent stimuli to make them, by making the conflicts between the auditory and visual stimuli, and we can measure their behavioral choices in these test trials after the learning. So we established 3 different systems, behavioral systems, for test this audiovisual discrimination ability in the mouse. One is in the head fixed condition, so the mouse head is fixed. So in the stationary mouse, you present the static stimuli in front of the mouse and measure their go, no-go, licking behaviors. And we also train the animal in the T-maze. In the T-maze, mouse should navigate freely and see the stimuli in the front to make the left or right choices. And as an intermediate system, we devised the treadmill system for the mouse. So the mouse head is fixed, and it can move their body. It can show the locomotion on the treadmill when they want, and we can present the auditory and visual cues either in the stationary condition or locomoting condition in this mouse. So we train the animal in these 3 different systems successfully and measure their behavioral choices under the congruent and incongruent audiovisual stimuli. So we published this story like 2017 in Neuron, and at the time, preview from the Dan Polly is published together, and this figure is well summarizing our finding. First, behaviorally, we found that the head-fixed mouse showed the auditory dominance decisions in the audiovisual conflicts. And then we traced the brain region that integrates the auditory and visual inputs in the mouse brain anatomically. So we did the anterograde tracing and retrograde tracing and found that the posterior parietal cortex received similar amount of inputs from the visual cortex and the auditory cortex in the mouse brain. And also we found that among the cell types in the posterior parietal cortex, PAV1 passive inhibitory neurons in the PPC receive stronger inputs from the auditory cortex than the visual cortex. So it can exert the feed-forward inhibition in the posterior parietal cortex. And we also did the electrophysiology in the PPC in the awake animal, and we measured the neural responses to the auditory stimuli, visual stimuli, and audiovisual stimuli. And the neurons that show the strong visual responses in the PPC showed the suppression in their activity when we gave the auditory stimuli together with the visual stimuli. So this physiological phenomena explains the auditory dominant behavioral decisions in the animal through this inhibition in the PPC. To prove that, we performed the mucimal inactivation and optogenetic manipulation of these circuits. So specifically, we targeted the PV neurons in the PPC, and optogenetic activation of PV neurons enhanced the auditory dominant decisions in the behaving animal. When we inactivate the PV neurons in the PPC, this auditory dominance switched to the visual dominance in the behaving animals. So by these findings, we propose a simple circuit, feed-forward inhibitory circuit in the PPC that mediates auditory dominant decisions in mice under the audiovisual conflicts. So these PPC neurons, there are PV passive inhibitory neurons that receive the stronger inputs from the auditory cortex, and that can suppress the visual responsive neurons in the PPC, which can mediate the auditory dominant decisions by suppressing the visual representation in the PPC. Initially, I was interested in the visual processing in the visual cortex, try to understand the microcircuits and neuromodulators in the visual cortex. And then recently we found that the posterior parietal cortex is important for the multisensory integration, especially resolving the conflicts between the auditory and visual inputs. And then we also recently found that the frontal cortex, such as the anterior cingulate cortex, is very important for deciding the go, no-go actions based on the sensory information. It can mediate the gated feed-forward inhibition to release the action decision by receiving the sensory inputs from the visual areas. So this is current series of our research. And I'd like to thank our lab members, especially the first work is done by Yu-Hyang and Jaeyeon. They are now postdocs in the US. And the second part, which is not published, is mainly done by Il-sung, who's now a graduate student in my lab. So thank you very much for listening.
Okay, thank you very much for that absolutely beautiful talk, an amazing dissection of a circuit with fascinating implications. We probably have time just maybe for a quick question, if there are any.
Hi, so amazing work. I was wondering, you showed all of this circuit that generates auditory dominance. Do you think there is a similar circuit to generate visual dominance, or is the visual like the default? And if it is it because this is a more dominant sense in mice, or what do you think about that?
Yeah, I think that's a very interesting and important question. So we mainly focus on the posterior parietal cortex that is important for the auditory dominant decisions, but we know that these auditory cortical and visual cortical outputs are integrated in other association cortical regions, and we also think that there must be some cortical region that is more important for the visual dominant decisions. So we are now trying to dissect the circuits further in the mouse brain as well.
Thank you.
Thank you.
I guess just to add on to that, I've got a quick question, which is I wonder why you think, why has it evolved this way? Why do you think that locomotion shifts it from, away from auditory dominance to visual? Is there some, evolutionary advantage? Is there a problem with the auditory perception when they're moving? What do you think?
Yeah.
Speculate.
So why is always a hard question. I don't know why the nature have this kind of phenomenon, but I guess when the animals start to use more visual information, when they are navigating in the environment, so usually with the light information, when we are moving around, we can see the cues better. And in that conditions, maybe our brain naturally make the circuits ready for more reliably deciding their actions based on the visual cues rather than the auditory cues, which can be maybe more distorted in the navigating conditions. So the behavioral states may modulate this further in this multisensory processing in the brain. But we have to see whether it's also conserved in the human brain, which is totally different from the mouse brain. Yeah.
Excellent. Okay, I think we should move on. So let's just thank Dr. Lee again for a really incredible talk.